Glucose is completely absorbed in PCT along with sodium. This reabsorption of glucose occurs along with reabsorption of sodium by symport or co-transport mechanism. Glucose and sodium bind to a common carrier, that is SGL2, that is sodium glucose transport 2 in the luminal membrane. Glucose is carried into the cell as sodium moves down its electrical and chemical gradient. The sodium is then actively pumped out of the cell to lateral intercellular spaces and interstitium. Glucose combines with another carrier protein, GLUT2, on the basal surface of the same cell and moves into the peritubular capillaries by simple diffusion. Thus, glucose transport across PCT is an example for secondary active transport. So here you can see with the sodium and glucose, they are going to attach to the SGLT2, then they are reabsorbed from there through the GLUT transport. They are going to reach to the peritubular capillary and glucose is absorbed. So this is called as the secondary active transport, because the gradient for sodium is produced because of the primary active transport, that is sodium potassium pump. A variety of other substances are transported by secondary active transport via symports with the energy provided by active transport of sodium out of the renal tubular cells. These substances include some amino acids, lactate, inorganic phosphates, hydrogen ion, and chloride. Glucose transport in kidney is inhibited by fluorescein, a plant glycoside which competes with the glucose for binding to the carrier. Glycosuria is common during pregnancy due to the decrease in renal threshold and increase in GFR. The characteristic of glucose transport and its excretion. If you see the rate of filtration of glucose at glomerulus, that is equal to resting plasma glucose level into the GFR, that is 80 into 125, that equals to 100 milligrams per minute. Transport maximum. The substances reabsorbed actively from the renal tubules require some specific transport system. There is a limit to the rate at which solute can be transported. This limit is due to the saturation of transport systems involved when maximum solute is delivered to the tubule. When maximum rate at which substance is reabsorbed from the renal tubule is called as transport maximum for that substance. For example, transport maximum for glucose, that is TMG, is around 375 milligrams per minute in males, and it is around 300 milligrams per minute in case of females. Renal threshold value for glucose. The renal threshold for glucose is plasma level at which the glucose first appears in the urine more than the normal minute amounts. This renal threshold is calculated by TMG divided by GFR. Theoretically, the renal threshold would be around 300 milligrams per liter, that is 375 milligrams per minute TMG divided by 125 ml per minute, that is GFR. However, the actual renal threshold is about 200 milligrams per deciliter of arterial blood or 180 milligrams per deciliter of venous blood. This anomaly between predicted value and practical value can be demonstrated by a graph. The actual curve is rounded and deviates from the ideal curve. So this deviation is called as splay. It represents the excretion of glucose in urine before TMG is reached. And this is due to all the nephrons does not have the same tubular maximum for glucose, and below TMG levels, the interaction between carrier and glucose is not completely an irreversible process. So some glucose escapes reabsorption. And this is a diagram showing the plasma glucose levels and glucose reabsorbed. As and when plasma glucose level increases, the reabsorption also going to increase, but at TMG maximum, the glucose starts appearing in the urine. And the dotted line shows the uh, relation between plasma glucose and the glucose reabsorb, that it appears quite early compared to the uh, the theoretical curve. That is, renal threshold uh, occurs actual renal threshold occurs before the renal threshold, and this is called as splay.